the process of transcription. Transcriptional processes, whereas in case of both prokaryotes and eukaryotes is same. The only difference is post-transcriptional modification is present in eukaryotes and absent in prokaryotes, which means in prokaryotes directly mRNA is produced, but in eukaryotes first hnRNA, which can be undergoes processing and then converted into mRNA. So in transcription process, three significant stages are there, that is initiation, elongation and termination. First, initiation. First, initiation. This is double-stranded DNA. Three prime to 5 prime. 3 prime to 5 prime, and another 5 prime 3 prime DNA. This is 5 prime and 3 prime. The point where the transcription is initiated, it starts, is known as promoter region, and it can be initiated by an enzyme RNA polymerase. The holo enzyme having 5 subunits are present: 2 alpha, 1 beta, 1 beta prime and omega, and mainly it is initiated by sigma factor, and along with this there is sigma factor also associated. With the help of sigma factor, the transcription is initiated. So this is the region what we can call it as promoter. RNA polymerase directly unable to recognize the promoter, so until unless the help of sigma factor. So here sigma factor which helps to bind the RNA polymerase to the promoter region. So this is what we can call it as initiation, and this is a double-stranded DNA helix. Next, elongation. Second stage is elongation. Once initiation is completed, the RNA polymerase can move along the length of the double-stranded DNA. Can move along the length of double-stranded DNA. Five prime, 3 prime, and this is 3 prime and 5 prime DNA. While moving the RNA polymerase through double-stranded DNA helix, it breaks the hydrogen bonds present between the nitrogen base pairs, hence the strands will separate slowly. So this is RNA polymerase enzyme. As soon as initiation can be started, the sigma factor is dissociated. Sigma factor is dissociated. This is sigma factor which is already dissociated, and slowly this is what we can call it as mRNA. Next, elongation. RNA polymerase after reaching into the terminator region and the transcription is terminated. After reaching into the terminator region. Yes, this is 3' to 5' region, and opposite strand is 5' to 3' region. So now after reaching here, the RNA polymerase is completely dissociated. And here, like sigma factor, another factor which is involved, this is what we can call it as rho factor. The rho factor involved in the termination of transcription. Whereas in case of prokaryotes, single type of RNA polymerase is involved, but in eukaryotes, 3 different types of RNA polymerases are involved. That is RNA polymerase 1, RNA polymerase 2, and RNA polymerase 3. Yeah, this is both in prokaryotes and eukaryotes. The process is same. After completion of transcription, this mRNA can be produced in prokaryotes directly. This is 5', 3', completely a processed mRNA can be produced. But whereas in case of eukaryotes, first hnRNA can be produced, and again post transcriptional modification. So that can be discussed in the coming topic. And this is what we can call it as mRNA transcript.